cDNA library. First, we know the mean of cDNA. cDNA means complementary DNA. The complementary DNA containing needed desired characters. Only desired characters present in which portion of DNA. That DNA is also called desired DNA or cDNA. The cDNA libraries mainly helps in the storage of desired DNAs, desired cDNAs in the library of cDNA. So first we know the mean of cDNA and what is important of cDNA library, how to construct the cDNA library. Let's discuss about the complete description of cDNA library construction. First, we know the DNA, eukaryotic DNA structure. Every eukaryotic DNA containing two long polynucleotide chains. They are arranged in opposite manner. The long polynucleotide DNA, eukaryotic DNA shows introns, exons. Exons of eukaryotic DNA important for nucleic acids or nucleotides. The exons are the nucleotide sequence in nucleic acids which code for proteins, hormones, enzymes. So these codes of exons important from DNA, eukaryotic DNA. Introns, non-coding parts of DNA. Every eukaryotic DNA shows these two major parts. They are exons, introns. Exons means needed genetic code for proteins, for hormones, for enzymes, for any other secondary metabolic activities. But introns are non-coding sequences, but they are also helps in formation of new genetic code, but they didn't have any another functions about the synthesis of protein, synthesis of hormones, synthesis of enzymes. This type of eukaryotic DNA involved in the transcription with the help of RNA polymerase enzyme. If the eukaryotic DNA involves in the transcription with the help of RNA polymerase enzyme, it means the DNA makes in the transcription pro-mRNA. The pro-mRNA is also called heteronucleated or heterogeneous mRNA. This total transcription process occurs in the nucleoplasm only. Means the DNA present in the nucleus, the DNA involves in the transcription. The transcription process produce pro-mRNA. It is the pro-mRNA. The pro or pre-mRNA. Pre-mRNA contains, again here it is 5 prime, 3 prime. With these two ends, two important portions are identified in pre-mRNA or heterogeneous or heteronucleated mRNA. They are also exons, introns. So, heterogeneous mRNA or pre-mRNA or heteronucleated mRNA also contains introns, exons. We know mean of exons, mean of introns. Introns are non-coding regions of mRNA. Exons are the coding regions of mRNA. So exons are important portions for protein synthesis, synthesis of hormones, synthesis of enzymes, or any other secondary metabolic chemical. So exons are important. Introns are not important from mRNA. Before the pre-mRNA develop into matured mRNA, developed mRNA by the splicing process. Splicing process means elimination of introns from pre-mRNA. See, these portions, introns are eliminated. Introns are non-coding part of mRNA. Exons are coding part of mRNA. By the splicing process, wanted material, exons are binded and introns are eliminated from the mRNA, heteronucleated or heterogeneous mRNA. So heterogeneous mRNA develop into matured mRNA. That matured mRNA contains only exons portions. So this exons portions of mRNA contains 5 prime, 3 prime. Between these 2 primes, coding sequence are present. So it is the matured mRNA developed from pre-mRNA by splicing process. Splicing process mainly helps in elimination of unwanted, non-genetic code, non-coding regions from heteronucleated mRNA. Only coding sequences are binded to make one matured mRNA. The matured mRNA is present in the nucleoplasm. If we observe that nucleoplasm, oligoprimers, oligo-DT primers are added on the mRNA at 3' of end. Oligoprimers containing deoxythymidine. T indicates thymidine. Deoxythymidines are repeated. See, we observed in this prime, oligoprime, T. T means poly-T. Poly-T means poly. More number of thymidines are present. The thymidines are added on the matured mRNA at the 3' of mRNA. If we added oligoprimers on mRNA at 3', we used reverse transcriptase enzyme. The reverse transcriptase enzyme helps in synthesis of cDNA from mRNA. It synthesizes the DNA from 5' to 3' direction. So finally, the matured mRNA synthesized new cDNA copy with the help of reverse transcriptase enzyme. The reverse transcriptase enzyme mainly isolated from retrovirus. Retrovirus containing this type of enzyme, the reverse transcriptase enzyme. So that type of reverse transcriptase enzyme is isolated from that viruses, retroviruses, and it is used for the making of cDNA from mRNA. If we added oligoprimers on the mRNA and we use reverse transcriptase enzyme, it synthesizes new cDNA copy from mRNA from 5' to 3' direction. Later, at end of these making of cDNA from mRNA, finally we observed hybrid mRNA DNA strand will present in the nucleoplasm. Why we call a hybrid mRNA DNA means half of the genetic material belongs to mRNA and remaining half of the genetic material belongs to DNA. So this complex containing mRNA and DNA genetic material. So this portion is also called, this part is also called hybrid mRNA DNA. After end of these synthesis of cDNA with the help of reverse transcriptase enzyme, we used hydrolysis process of mRNA. Hydrolysis mainly helps in fragments of mRNA from hybrid mRNA cDNA, mean elimination of mRNA strands from cDNA. This process is called hydrolysis. Before the using of hydrolysis, hybrid mRNA cDNA are present. If we use the hydrolysis process, the mRNA cleaves into fragments and it leaves from the cDNA strand. Finally, we observed only single strand DNA. The single strand DNA is also called complementary single strand DNA. the single strand DNA, 3 prime, 3 prime turn into hairpin loop-like structure with the help of reverse transcriptase enzyme and also with the help of nuclease enzymes. The nuclease enzymes and reverse transcriptase enzymes are mainly helps in the formation of hairpin loop. 3 prime of cDNA turn into hairpin loop-like structure. If the hairpin loop-like structure is identified on the cDNA 3 prime, if we apply DNA polymerase 3 enzyme on this cDNA strand, so the DNA polymerase 3 synthesize new secondary strand of DNA with the help of DNA polymerase 3. Finally, the DNA polymerase 3 make the second strand of cDNA to make double-stranded DNA. The double-stranded DNA containing one hairpin loop-like structure at one end. The hairpin loop-like structure is eliminated or deleted by S1 nuclease enzyme. With the help of S1 nuclease enzyme, the hairpin loop-like structure is eliminated. Finally, we observe in the nucleoplasm pure double-stranded cDNA. In this cDNA, there is no introns, only exons are present. This type of DNA is involved in the transcription. It develop into complete matured mRNA. That matured mRNA develop into needed proteins, needed hormones, needed material to cell. So this type of DNAs are synthesized by reverse transcriptase enzyme. And this type of cDNAs are stored in the libraries. So it is the total process of how to make the cDNA, how to construct the cDNA. Finally, at the end of the making of cDNA, this type of cDNAs are stored in the libraries. That libraries are called cDNA libraries. So before the storage, the cDNA libraries, cDNAs are treated with restriction endonuclease enzymes. The restriction endonuclease enzymes helps in the sticky ends, formation of sticky ends on the cDNA, and that same restriction endonuclease enzymes also used in the selected plasmids. So that plasmid and the cDNA make new rDNA. That rDNAs are also called chimeric DNAs. The chimeric DNAs are insertion into E. coli bacteria with the using of calcium chloride solution, and finally that bacteria are helps in safely storage of cDNA. It is the storage of cDNA libraries. It is the construction of cDNA libraries in the genetic material ways are used in the biotechnology process.